Hello, my name is Patrice Johnson, and I'm a clinical pharmacist and project manager. I want to start off by thanking Diabetes Sisters for having me present this on-demand webinar to you today. Over the last several months, I've studied and evaluated data to better understand COVID-19 and the new vaccines. Like you, I've had people very close to me develop COVID-19, survive it, and unfortunately die from it. I'm committed to helping you to make an educated decision about receiving the vaccine. Today, we're going to review some of the terminology used when talking about SARS-CoV-2 virus, how vaccines work, specifics on how the COVID-19 vaccines work, and what to expect when you get the vaccine. So let's start by talking about viruses, their structure, and how they work. So first of all, viruses are very simple structures. They contain really just three parts: the genetic information right here in the center, then there's a surrounding envelope to protect that genetic information, and then there's a protein coating. So going back to that genetic information, think of that as its recipe. One thing that's unique about viruses is that they cannot replicate on their own. They need our host cells to replicate. So viruses multiply by unlocking the door to our cells. They then take over our machinery and make more of the virus. Remember, viruses can't replicate on its own. So earlier I talked about that genetic information that's at the center of the virus. Think of that like a recipe. It's a recipe to make more of the virus. What viruses do is they use these spike proteins almost like hands to unlock the door to our cells. So think of it like a virus doesn't have its own kitchen or its own groceries to make a recipe. It's got to unlock the door to our house, use our groceries, our blender, our stove to make more of itself. These spike proteins around the image, again, are what unlocks the door. Now, you've probably heard the words antigens and antibodies more than you'd ever care to hear in the year 2020. So let's take a minute to define them. An antigen is a substance that causes an immune response by the body. Antigens are found on the surface of the virus and are unique to that virus. So understand that the antigens that are unique to SARS-CoV-2 virus are not the same as the antigens for the H1N1 flu, or the same as the shingles virus, or the same as the chickenpox virus. Each virus is very unique and has their own antigens. So when a virus enters the body, our body evaluates the virus to determine, is it foreign, is it harmful? Once it does that evaluation, it signals our immune system to go at that virus. And so one of the things our immune system does, among many other things, is it will create antibodies. So represented here in this illustration on the left, we've got a virus, and around the edge of that virus, we've got our spike proteins. And so as our body recognizes that this virus is foreign and harmful, it's going to begin to make antibodies. Those antibodies are going to counteract that virus. Now, what's unique about antibodies is they're very specific to the antigen. And so what you see here in this illustration is the antibody, that Y shape or wishbone shaped object, is actually going to cover part of that spike protein. So going back to that analogy I made earlier of viruses having these, covers, like the spike proteins almost act like hands to unlock the door to our cells. Antibodies inactivate those hands. So the virus can no longer latch onto our cells because they can't open the door to get in to use our machinery to make more of the virus. And that's why antibodies are so important, because they literally counteract that protein spike, that spike protein, so that it cannot latch on to our cells, open the door to our cells, and create the replication of the virus. If it can't replicate the virus, it can't make us sick. We want our immune systems to have the strongest possible response to a virus. The illustration here shows exactly what we want to see the immune system do. Here we've got a virus, that's what the red circular structure is, and then we've got antibodies coming at that virus. And those antibodies are attaching to those spike proteins, inactivating their ability to unlock the door to our cells. Therefore, we can't become infected with the virus. So when you hear of therapies like monoclonal antibodies or convalescent plasma, which has those antibodies in it, that's a therapy that's used to help boost our immune system because it's providing us with the antibodies that our bodies have not made yet. Before I explain details on the COVID-19 vaccine, I'd like to share with you a bit about some of the different vaccines that have already been in use for years. Prior to development of mRNA vaccines, there were four main categories or methods for making vaccines: weakened or live vaccines, inactivated, subunit or conjugate, and toxoid vaccines. And although the first two COVID vaccines will be mRNA vaccines, which we'll discuss shortly, there are several other COVID vaccines that are currently in trials that are made using some of these existing methods that I'm going to talk about. So I want you to be able to understand what the differences are. So I'll just take a few minutes here to explain the different methods in making vaccines. So first, a weakened vaccine is one that's been treated in a way to reduce its virulence, yet allow it to still be able to provoke an immune response. So in a weakened vaccine, the virus is isolated and then it's grown in a way so that it makes it no longer good at replicating itself. I like to compare this to copying a document on a copier that's running low on toner or ink. As you make more copies, your print gets lighter and lighter. Although the lightest copy is still a sheet of paper, it shouldn't be used to make more copies. So that's how a weakened vaccine works. It still contains the virus, but that virus can no longer replicate at a level that can make you sick. It can, however, mount an immune response, which allows your body to build antibodies that would recognize that virus if you were affected with it in the future. Inactivated viruses have been treated with heat or a chemical, then purified to render them no longer able to replicate themselves, yet still cause an immune response. That would be like your flu vaccine, your rabies vaccine, your polio vaccines. Those are great examples of inactivated vaccines. Subunit or conjugate vaccines use the antigen. Remember, antigen is a piece of the surface of the virus. And so with subunit or conjugate vaccines, they use just a piece of that outer surface, a piece of the antigen, but not the genetic info, the part, that recipe that's inside the membrane of the virus. That's what's used to make a subunit or conjugate vaccine. This allows our immune system to recognize the antigen, build the antibodies, and prepare us if we come into contact with that virus in the future. Some of the other vaccines that are currently in trials for COVID-19 are going to be conjugate vaccines. So you'll see these show up more later in 2021. A great example of a subunit or conjugate vaccine is the shingles vaccine, which is highly effective. Toxoid vaccines are created to protect us against dangerous bacteria like diphtheria and tetanus. A toxoid vaccine takes the dangerous toxin or poison that's secreted by the bacteria, treats that toxin with heat or chemical to inactivate it, purifies that, and then places that toxoid in a vaccine to elicit an immune response. So as you can see here, there's a common theme. Every vaccine that's been created is designed to have our immune system respond and build antibodies. Now let's talk about mRNA vaccines. To help you understand this, let's first take another look at the virus. We've got the genetic information or the recipe to make more of the virus. We've got the membrane, the outer membrane, and then we've got that protein coating or the spike protein. Now, in making the vaccine, what they've done is taken just a component of the recipe, just a part of the genetic information, but just a part of that recipe is used to make the mRNA vaccine against COVID. The part that they've used is the part that makes the spike protein. By placing that recipe for the spike protein into a vaccine, our cells are given the instructions on how to make just the spike protein. Once our cells make the spike protein, then our bodies will recognize it as foreign, potentially harmful, and our immune systems will build antibodies against it. So again, notice that the vaccine only includes that part of the recipe to make the spike protein. It doesn't include the entire recipe. So by just having the instructions or the recipe to make that spike protein, again, our cells are going to make the spike protein. Our immune system will assess that spike protein, recognize it shouldn't be there, build these wonderful antibodies that will be available to us if we ever do come into contact with the SARS-CoV-2 virus. So, can I get COVID from getting a vaccine? No, you cannot get COVID from getting the COVID vaccine. Remember, the mRNA vaccines only contain the instructions to make the spike protein. They do not contain instructions to replicate the virus, simply the spike protein. Now, it is important to understand, though, that vaccination does not mean immediate immunization. All vaccines either present us with the antigen or teach us to make the antigen in the case of the mRNA vaccines, which then cause our immune system to spring into action and build antibodies. This takes time. So if you're exposed to SARS-CoV-2 virus right before you get the vaccine or shortly after you get the vaccine, yes, you could still develop COVID-19 disease. Depending on the vaccine you get, it will take about 7 to 14 days to build your strongest immunity after receiving the second injection, whether it's the Pfizer or the Moderna vaccine. I'm often asked if there were shortcuts taken to get this vaccine made. The answer is no. For that reason, I want to take a minute to go over the process. In developing a vaccine, the first step is to identify the antigen, which was done back in January 13th of 2020. mRNA technology was chosen by at least 2 drug companies as the most effective way to create a vaccine, and they began developing a product. Testing was done in animals, and then applications were filed for the test to be able to be done in humans. Phase 1 trials were conducted in 20 to 100 people, and those trials are done to evaluate basic safety and identify the most common reactions with receiving the vaccine. In phase 2 trials, which in some cases were started just as phase 1 trials are wrapping up, those trials enroll several hundred participants. In some cases, there were a few thousand. And those trials are used to determine the dose and the frequency of the vaccine, and then also what to expect when receiving the vaccine. Should the dose be 0.3 milliliters? Should it be 0.5 milliliters? Should it be given day 1 and day 10 or day 1 and day 21, and so on? So that's really ironed out in phase 2. And then in phase 3 trials, it's opened up to a much larger group of people to start to see if there's any other effects that we didn't see in the smaller populations that were studied in phase 1 and phase 2 trials. Now, because COVID-19 became so prevalent across the world, we knew early on in this year that this virus was adversely impacting people of color, people of advanced age, people with comorbid conditions. We knew it was dangerous to these groups of people. And so in doing these studies, it was important that we assess the ability of this vaccine to protect these very vulnerable populations. And so therefore, in the phase 3 trials for both the Pfizer vaccine and the Moderna vaccine, People in these vulnerable age groups, ethnic backgrounds, and health statuses were included in phase 3 trials. And I'm really happy about the results in those populations. Earlier, I talked about how viruses unlock the door to our cells. They hijack our machinery and use our machinery to replicate itself. I also said that as more cases of COVID-19 developed, as more cases popped up, we quickly realized that the severity of COVID-19 disease caused by the SARS-CoV-2 virus was far more detrimental in certain populations. You may wonder why that is. Well, it goes back to what I said earlier about how a virus works. The spike proteins on the SARS-CoV-2 virus have a particular affinity for some very important cells in our body. These specific cells line our lungs, our arteries, our kidneys, and are involved in the regulation of our blood pressure, just to name a few. So you can imagine that anyone that has a disposition or predisposition to challenges with these organs would be severely impacted by this disease. Therefore, having patients with diabetes, cardiac disease, lung disease, and obesity in the trials helped us to learn how we can protect this vulnerable part of our population. The trials were able to demonstrate 100% effectiveness at preventing serious illness from being infected with the SARS-CoV-2 virus. This is tremendous. So in addition to knowing that the vaccine was tested in people with comorbid conditions, it was also important to have participants in the trials that were racially diverse and of advanced age, because we also know that people from those diverse backgrounds and higher in age tended to have some of the really negative consequences from developing severe COVID-19 disease. So in the Pfizer trial that contained over 43,000 people, 30% of the participants were racially diverse, 10% African American, 13% Hispanic, over 45% were over the age of 55. And then for Moderna, about 25,000 people in the trials, 37% racially diverse, 10% African American, 20% Hispanic, and then 23% were over the age of 65. So again, with what I've shared so far, you can be sure that these vaccines were tested in people with comorbid conditions, of advanced age, and in racially diverse populations. Another question that I'm often asked is, what is a EUA? An emergency use authorization, or EUA for short, for a vaccine is based on the need to use a vaccine quickly to save lives during a public health emergency. At the time that I'm recording this webinar, over 330,000 people have died due to COVID-19 disease. To put that in perspective, it would be equivalent to the entire population of the city of St. Louis or the city of Cincinnati. So this is absolutely a public health emergency. An EUA is a shorter process, but no steps are skipped in the safety evaluation. And so as I shared earlier, all the things that we normally do in a vaccine approval process were done. Simply the FDA, what they do to grant an EUA is to compare or assess the vaccine's known and potential benefits and be sure that those known and potential benefits outweigh the known and potential risk. And in this case, that was absolutely the case. Having 94 and 95% effectiveness in preventing the COVID-19 disease, and then 100% effectiveness in preventing severe COVID-19 disease is just incredible. In addition to the FDA's review process, there are two separate advisory boards that review the data that's presented by the drug companies and from the trials. They are the Advisory Committee for Immunization Practices and the Vaccines and Related Biological Products Advisory Committee. Both of these have independent scientists, independent physicians that review the data and make recommendations. So just a quick comparison on the two COVID-19 vaccines that are available as of today. The Pfizer product is a zero point 0.3 milliliter dose. It's given two doses, 3 weeks apart, so day one and then 21 days later for the second dose. It was studied in people as young as 16, therefore it is approved for use in people as young as 16. It does require ultra cold storage, and the protection that is talked about for the Pfizer vaccine really comes about 7 days after that second step of dosing. And so if you understand that you get your first dose day one, second dose 21 days later, about 7 days after that, that's where you get that real immunity because it's given your body time to build those antibodies. For the Moderna product, it's a zero point 0.5 milliliter dose. Two doses are given 28 days apart. It's approved for people 18 and over. It was not studied in people younger than that, therefore, it's not approved for people younger than that. And lasting protection with the Moderna vaccine is about 7 to 14 days after that second step of dosing. So it is important for both of these vaccines that you receive both doses and that you stick with the same vaccine. So if you started with the Pfizer vaccine for your first dose, it's important that you continue with the Pfizer vaccine for your second dose. The other thing that I want to mention is that there are more vaccines that will be coming to market in 2021. We've got 7.7 billion people to immunize. And so, we can't depend on just one company to make that many doses. So remember some of the things I shared with you on how other vaccines are made, because there will be more than just mRNA vaccines available to us for protection against COVID-19 in the future. So who should not get the vaccine? Well, there's two special populations that would be of concern. One, patients who have had a severe allergic reaction after a previous dose of COVID-19 vaccine, and two, patients who've had a severe allergic reaction to any ingredient of this vaccine. For that reason, immunizers are required as part of our standards to have EpiPens available in the event of anaphylactic shock. Also, for people who have had any history of allergic reactions that are that serious, the monitoring for those patients is to be extended to 30 minutes. It's normally 15 minutes post immunization or post vaccination. For people who've had an allergic reaction in the past, it would be 30 minutes. But again, these would be the two populations that are of concern. In order to get on the other side of this pandemic, we're going to need vaccination rates at the very minimum to be 70%. Ideally, we want them over 85%. I've pulled this slide from a journal, an Australian journal, to help illustrate why high vaccination rates make a difference. So in this first row, let's just take a look at the impact of having two people who are infected with SARS-CoV-2 go into a large group. When we look over to the right, we see how many people just those two people can infect. It can be, 10, 12 people in that large group that were closest to them. When you have a few more people, say 10% of the population that has received a vaccine or they've had previous exposure to a disease and so they've got some immunity, when you take those two infected people along with 5 or 6 people that have immunity, the impact is not as bad. It's a little bit better in terms of the number of people that get infected. What we're shooting for is to have a highly vaccinated population, so there's a lot of immunity. That way, if you take those same two people and put them in a large group, they're less likely to infect so many people, and therefore we can get on the other side of the pandemic. The other benefit of this is that our healthcare system will be better able to handle when one additional person is infected as opposed to 30 additional people. So high vaccination rates is incredibly important. It's also important that these high vaccination rates happen all throughout our country. It won't work if Minneapolis is well vaccinated, but Chicago isn't, or New York is well vaccinated, but Rochester isn't. And so this is really, really important. So I'm hoping that this helps you to understand why that vaccination rate number needs to be so high. So, what should you expect when you get the vaccine? Well, first of all, the vaccine's not going to give you COVID-19. Got to say that again to remind you the vaccine cannot give you COVID-19. However, what you can expect is that there may be some short term discomfort. There may be some fatigue, headache, muscle ache, chills, fever, and tenderness at the injection site after the vaccination. Now, usually you see that within about a day of receiving the vaccination, and these symptoms tend to dissipate within 72 hours. So they show up, within a day, maybe last a day, maybe it last 2 days. So really, 72 hours after you've had the vaccination, all of that goes away and it goes away on its own. In the trials, less than 2% of the participants in the trials thought that these symptoms were even significant or considered them severe. So again, less than 2% of people in the trials thought these were severe, and no one required hospitalization for, these symptoms. The other thing that I think that's important to understand is that these symptoms are associated with an immune response. It's a sign that your body is doing exactly what it needs to do, which is make antibodies to create those antibodies so that in the future, if you come in contact with the SARS-CoV-2 virus, you're protected. So these are symptoms associated with an immune response. They're normal, they're common, and they should be expected. So I'll take a couple of minutes to answer some common questions. One is, when and how long will I be protected? Since the virus has only been around for a little less than a year at the time that I'm recording this webinar, we just don't know how long antibodies last. We're currently studying this, but again, since the virus is so young, it's only been, around for a little less than a year, we don't know how long antibodies last. We believe they last at least 3 months, but we're not sure how long they last. And so this may be the type of vaccine that we have to get every year, maybe every couple of years. Will I still need to wear a mask? Yes, we will need to continue wearing masks. We will need to continue social distancing, washing our hands, hand sanitizer, all of those things until we get to a point where most of our population has been vaccinated. One of the things we don't know yet is whether the vaccine protects us from an infectivity standpoint. What I mean by that is even though you may have gotten the vaccine, you've gotten your second dose, you've now built immunity, we don't know if you come into contact with someone who tests positive for COVID, you might still be a carrier even though you wouldn't get seriously ill from it. Because of that, we really need to get to high vaccination rates and get to a point where the risk of infecting other people is very, very low. So for now, yes, you will still need to wear a mask. What if I've already had COVID-19? You are still a perfect candidate to receive the vaccine. Unless you are actively fighting a COVID-19 infection, and I mean, you're in that part where you should still be quarantined, I would recommend that you get the vaccine. Is it safe for pregnant and nursing women to take the vaccine? Absolutely. The American College of Obstetrics and Gynecology has put together a very nice statement on their website explaining that it is safe and that pregnant and nursing women should not delay in getting the vaccine. And then where should I look to get accurate information? So there's a CDC website, there's a Pfizer website, and there's also a Moderna website that have a lot of information for both healthcare professionals and for the general public that I found to be very helpful. So on this next slide, I'll show you those websites. I've listed here a few great references and websites for additional information on vaccines, and specifically the Pfizer and Moderna websites for information on the COVID-19 vaccines. What I also want to add is that the CDC has an incredible amount of information, not just on COVID-19 vaccines, but on what to expect with other vaccines, how vaccines work, other information about, plenty of other diseases. So really the website is very well done, very easy to navigate, easy to search through. So I'd highly recommend you start there. Getting on the other side of this pandemic, stopping this pandemic, is going to take us continuing to protect ourselves and others by using every tool in the shed. We've got to continue wearing a mask. We've got to continue to social distance, wash your hands or use sanitizer when hand washing is not available, quarantine when required, and get vaccinated, both shots. So that's going to be our best defense in getting on the other side of this. So I want to thank you for taking time to listen to this webinar today. We appreciate your support of Diabetes Sisters, and I truly hope that you found this information helpful. Thank you and stay well.